Good morning, everybody. We're really delighted to see such a well-attended session near the very end of the conference. We hope that you'll benefit from the program that we've tried to put together, really dealing with a number of issues surrounding gene editing, gene delivery, and the overall issue of delivery as well. So I'm joined by Hilary Valley from the CFTR laboratory, who'll also be giving one of the talks today. And no disclosures here. So essentially what our objective is, if we think about the airway, is if we start with essentially a CF airway, where there's little to no production of functional CFTR protein, and we have a sort of a thick dehydrated mucus. The overall concept is either through sort of luminal delivery, or perhaps through systemic delivery of getting in either genetic material that will encode for CFTR, or perhaps to deliver editing modalities that can restore gene protein expression and function. And so the idea over on the right-hand side here, as we sort of look at the blue cells that now have the CFTR channels again, is the idea to be able to get some percentage of cells in the airway that are now expressing functional CFTR protein sufficient that we now have good hydration of the mucus again, and then we have the ability of the cilia to basically clear pathogens out of the airway. So in terms of, we think about how we can accomplish this, it could either be through sort of delivery of messenger RNA or perhaps of viruses such as AAV that will deliver the genetic material, or it could be the goal of gene editing, which is really to target the chromosomal DNA to actually correct those mutations. And in fact, in today's presentation, we're going to be looking at several modalities of how we may be able to restore CFTR expression with a function with a sort of a focus primarily on editing. So this is just showing here an outline of possible. Oh, thank you very much. Is that better? Okay, thank you. I'm sorry. Very good. I could hear myself quite well, but I guess that's not the point today. So the idea here is if we look at the left-hand schematics, we're really looking here at the full CFTR genomic sequences together with the sort of the star, the asterisk, sort of indicating a mutation. And it can be a stop codon, it could be a missense mutation, for example. And so the idea is through sort of integrating vectors is through accomplishing, if you will, sort of random integration somewhere else in the genome where we leave essentially the CFTR locus unmodified. And the idea is to restore with appropriate promoter sequences and perhaps insulators to in fact achieve expression of the full CFTR cDNA. Another possibility is now this is sort of moving into sort of gene editing. The second line is really looking at the issue of sort of a safe harbor approach. And there have been some reports in the literature looking at this where you use some kind of sort of editing methodology to target the integration into a very specific locus. Again, you deliver the regulatory sequences, and this is another modality that is possible, still leaving intact the mutant CFTR locus. The final two are what we think more typically of sort of an endogenous editing approach. The third from the top is essentially site-specific correction, and which is really to replace the mutant sequences either through CRISPR-Cas9, it could be through base editing, it could be through prime editing, a number of ways in which one could actually do site-specific correction of mutations. And again, so the idea with the endogenous correction is the ability to sort of retain the normal endogenous regulation of expression so that we can recapitulate in cell types either no expression, for example, in ciliated cells, high-level expression in ionocytes, or then by targeting basal cells and differentiate into, for example, secretory cells, basal cells, is to sort of retain that level of expression as well. And finally, the bottom one is sort of this superexon approach, which is to try to sort of independent, sort of a mutation agnostic manner, independent of the mutation that's downstream, to target sort of a partial cDNA that has a splice acceptor, has polyadenylation sequences, with the idea of being able to restore the complete functional activity. And there have been a lot of talks and also in this session as well, we'll deal with this issue. So if we look at the speakers, so what we've tried to do is a significant amount of sort of developments in the field have occurred in academic laboratories, and we have representatives from 2 universities here. There's also a lot of work in terms of, for example, the CFFT laboratory, that's represented in our first talk by Hilary Valley. And then also a significant amount of advancement, and particularly taking these things into clinical trials, has been achieved through more industrial corporate organizations. And we have Jeremy Duffield, who will be representing Prime Medicine here. We have Sriram Vidyanatham from Nationwide Children's, who's going to be presenting on sort of how does various variations that exist naturally within the genomic sequences, both on target as well as off target, affect how we would design these editing strategies. And finally, Philip Santangelo from Emory is really going to talk about this issue of delivery. Our ability to achieve either gene delivery or gene editing depends on being able to get the reagents of interest into the appropriate cells where they can accomplish either their delivery, the expression, integration, or editing. So what I'd like to do is sort of hand this off to Hilary at this time. Basically, we have a good amount of time to work with. We're going to go with 20-minute presentations, 5-10 minutes for Q&A, and we'll just see how things go. If we have a significant amount of time at the end, we'll have a panel discussion, but we hope really to have some very robust Q&A following each of the presentations. So I want to thank you for being here and welcome Hilary as our first speaker.
Thank you, Brian. All right. Good morning, everyone. I am very excited for this session, and I'd like to thank my co-chair, Brian, and the Program Planning Committee for the opportunity to be a part of it. Today, I'm going to be talking about a therapeutic strategy for CF known as the Superexon approach. And I have nothing to disclose. So hopefully by this point in NACFC, we're all aware that there are many mutations that cause CF, and these are located all throughout the CFTR gene. I really like this diagram from Sick Kids Canada that shows this graphically. Up at the top, we have the 27 exons and 26 introns of CFTR. They also diagram with these individual lines, the many missense, nonsense, and other types of mutations. And these are located all throughout the CFTR gene. So as we start to think about genetic therapy approaches for how we can correct these mutations, we have a number of options. Option 1 is the most obvious, where we can use an individual genetic therapy to provide benefit for one mutation. And this is really promising for mutations such as G542X, the most common mutation for which there is currently not yet an approved therapy that treats the basic defect. However, because there are so many mutations in CFTR, we also know that this is not a feasible path to correct every mutation. There are other strategies where one genetic therapy could provide benefit for several mutations. For example, in exon 12, one could correct all of the mutations within exon 12, and this is also a really exciting avenue. The last strategy is the Superexon strategy, which could provide benefit for really most of the CF-causing mutations. And on the next slide, I'm going to diagram more about what that is. But here I want to just mention that the Superexon approach will probably not be the first editing approach that makes it to the clinic. Delivery of large cargo and insertion of that cargo into the genome remains a challenge, although we've seen a lot of progress on that in the past few years. However, we want to continue to push forward these therapies that can repair many mutations with one therapy, because ultimately, that's where we want to be. So what is a superexon? Very broadly, a superexon is a partial cDNA of the CFTR gene inserted into the native CFTR locus. And there are many different size options and insertion locations for superexons. Brian Davies, Sriram Vidyanatham when he was a postdoc in Matt Porteous's lab, we'll hear from later, Susan Burkett and others have all made different size superexons and contributed to our understanding of CFTR superexon biology. Today, I'm going to be talking about two different size superexons. And importantly, I'm going to be focusing on superexons inserted into CFTR introns. And the reason for this is because given the mechanisms of current gene editing technologies, we think that intronic insertion of a superexon is likely to be safer and offer more targeting flexibility. This diagram shows a CFTR superexon consisting of exons 2 through 27 inserted into intron 1. Now, this construct also consists of a 3' UTR and polyadenylation that's not what I wanted to do, sorry. Polyadenylation signal sequence to mark where the superexon ends. And because it's being inserted into an intron, it also contains a splice site or a splice acceptor. Now, a superexon can theoretically provide benefit for all people with CF with one or more of their CF-causing mutations downstream of the insertion site. So a superexon inserted into intron 1 could provide benefit for over 99% of people with CF in the US. Now, before I dive into some of our superexon data, I want to outline what our goals around superexons are at the CFFT lab. Our goals are to develop and optimize a blueprint for an effective exon 2 through 27 superexon inserted into CFTR intron 1. And we want to share this blueprint with other groups so that they can harness their unique gene editing technologies and delivery modalities to insert a validated and optimized superexon. Really what we're trying to do is optimize the cargo. And I hope that by the end of this talk, you'll appreciate that it is important to optimize the cargo. So actually, first we started with a smaller superexon consisting of CFTR exons 23 through 27 inserted into intron 22. This superexon could provide benefit to about 15% of people with CF, including all of those with mutations downstream of the insertion site, which includes but is not limited to 1282X in exon 23 and N1303K in exon 24. And we did this using conventional CRISPR-Cas9 based approaches, and we delivered the superexon to 16-HBE GE1282X cells. Importantly, we also subcloned out those cells to isolated individual clonal cell lines with the superexon inserted and then characterized them. And we did this because, remember, our goal is focusing on the cargo. And what we want to do is we want to isolate the superexon function. We want to isolate the effect of that cargo and dissociate this from insertion efficiency. At the top, this is our first generation exon 23 through 27 superexon. It contains the very commonly used BGH polyadenylation signal sequence, as well as the native CFTR exon 23 splice acceptor site. The data here is electrophysiology data from a TEC24 assay, and 16HB14O- wild-type cells have robust CFTR function as expected and as demonstrated by the area under the curve. Again, 1282X cells do not have appreciable CFTR function as expected. And our superexon lines had about 5 to 10% of wild-type function. And initially, we were really surprised by this low level of functional rescue. So we did some sequencing and other characterization and developed this hypothesis for why we were seeing incomplete superexon rescue. This is what the genomic DNA looks like when a superexon is inserted. Importantly, nothing is actually removed from the genome. The rest of the gene is just pushed downstream when the superexon is inserted. And what we want to have happen is that we want polymerase to transcribe this and for the transcript to be effectively terminated, cleaved, and polyadenylated at the end of the superexon. And after mRNA processing, this will give us full-length wild-type CFTR. But what was actually happening is that we had transcriptional read-through, and we were continuing to transcribe the rest of the CFTR gene. And this means that during mRNA processing, there was a couple of splice options. Splicing could occur to the superexon as desired, which would give us full-length wild-type CFTR. Or splicing could bypass the superexon, which would recapitulate the mutant 1282X CFTR, which is obviously not what we want. So we hypothesized that, okay, well, if this is the splicing issue, then let's use a stronger splice acceptor site. So Brian Davies kindly provided us with a very strong synthetic splice acceptor. And so we put this into our superexon construct. And so that's diagrammed up here. It's the exact same as the first generation, except now it has a stronger splice acceptor site. And here we're now seeing about 20 to 40% of wild type rescue, which is an improvement, but still not where we want to be. We also realized that when we were trying to quantify the level of bypass splicing, that it was a bit difficult because we were doing this in a 1282X system. 1282X is sensitive to NMD, whereas wild type or superexon mRNA is not. So what we did is we switched to a model that was not sensitive to NMD, an N1303K model, put the superexon into this model, and again, we saw about 30% of functional rescue. So while the stronger splice acceptor was helping, it was clearly not enough. So in addition to the strong splice acceptor, we wouldn't necessarily need to have as strong of a splice acceptor if we were really effectively terminating our transcript. So we wanted to try a different 3' UTR and polyadenylation signal sequence. So in this third generation construct, we have both a strong splice site as well as the SV40 polyadenylation signal sequence. And now we're getting about 50 to 100% of wild type functional rescue, which is much more in line with what we were hoping to see. We've also been collaborating with Craig Hodges and Ron Conlon at Case Western Reserve University to look at superexons in mouse intestinal organoids and in mice in vivo. And I'm really excited about this. Craig gave a talk yesterday, which I hope all of you were able to attend, but in case not, I'll just give a very brief overview of just a small portion of the work that he showed. So what they did is they took the superexon that worked really well in the 16 HBEs, and they delivered this to mouse intestinal organoids that had the 1282X mutation. Now, CF intestinal organoids do not swell, and when they are corrected, whether it be with modulators or the superexon, then they swell. So do these superexons swell? Does this video work? Yes. Okay, good. Yes, they swell, which we were really excited to see. And Craig and his team also quantified this. And so this is the quantification of the swelling. So here we're looking at wild type organoids, superexon organoids with the mouse coding sequence or the human coding sequence. Human coding sequence, as well as conventionally corrected 1282X. And these all looked very similar to wild type and all looked very different from the starting material, which was the 1282X organoids. So importantly, this exon 23 through 27 superexon construct restored CFTR function in mouse intestinal organoids. So to briefly summarize part one, there are 3 parts. Our early work with CFTR superexons focused on a smaller exon 23 through 27 superexon construct. We initially saw low levels of functional rescue, only about 5 to 10% of wild type. But when we optimized the splice acceptor and polyadenylation signal sequence, we were able to improve that functional rescue by 10-fold. Importantly, the superexon construct also resulted in functional rescue in mouse intestinal organoids. So as an overall take-home, superexon genetic components, and by genetic components, I mean the splice acceptor, the polyase signal sequence, and other features of it, can have notable effects on superexon expression and functional rescue. We then moved on to our ultimate goal, which is a CFTR exon 2 through 27 superexon inserted into CFTR intron 1. And again, because we were focusing on the cargo, we isolated clonal cell lines that had the superexon inserted. The methods are a little bit different here. We're doing this in G542X cells now. And because the cargo for this is really quite large, for our in vitro proof of concept studies, we are using an adenovirus to deliver the superexon construct. So we were able to isolate 11 cell lines that had the superexon inserted. The Western blot data is shown on the right here, and the qPCR for 3 of the 11 is shown on the left. Now, as I hope you'll appreciate from the Western blot, that there is really robust functional rescue. In the first lane, we have 16HB14O- wild type cells, or wild type CFTR. In the second lane, we have G542X, so no CFTR is expected. And then the 11 clones have qualitatively very robust levels of functional rescue, and we were really excited to see this. We also looked at all of these clones by RT-qPCR, and for brevity, I only put 3 of them in here, but actually all of the clones had lower levels of steady-state CFTR mRNA than the wild-type control. So we were a bit surprised by that. We also looked at functional rescue, because we obviously want to look at function. That's a really important metric. And so I've quantified the data here, and the y-axis is the foreskin area under the curve. Again, wild-type cells have robust levels of CFTR function. G542X cells do not. And our 11 superexon clones had really robust functional rescue. Now, there were 8 clones that had 1 copy of the superexon inserted, and these are in black. And there were 3 clones that had 2 copies of the superexon inserted, and these were shown in purple. Now, 16 HBE cells have 2 genomic copies of CFTR. However, we refer to them as functionally monoallelic. And that's because during the immortalization process back in the 1980s, there's a complex insertion of SV40 into intron 6, which means that both alleles produce mRNA, but only one allele produces functional CFTR. Now, we had long hypothesized that if we put in a superexon upstream of that intron 6 site, that we would essentially functionalize the other allele. And this is exactly what we've done. And so in the clones that have 2 copies of CFTR, they should now have twice or more, if it's a really good superexon, levels of what you would see in the initial wild-type line. So with 2 copies of the superexon inserted, we saw about 261 to 327% of wild-type rescue, which was really exciting. So to briefly summarize part 2, we've demonstrated proof of concept for a CFTR exon 2 through 27 superexon inserted into intron 1. And we saw really robust functional rescue, about 76 to 161% of wild type, with 1 copy of the superexon inserted. However, despite high levels of functional rescue, mRNA abundance and stability, which I didn't have time to show, was reduced, suggesting possible altered mRNA stability or translational efficiency, kind of hand-waving a little bit here. And this is an area for continued study and optimization. Now, this is very much a working hypothesis, but what I think may be happening is that what we're lacking in mRNA stability, I think we're making up for in splicing efficiency. Now, normal wild-type CFTR has 27 exons in 26 splicing events. But we also know that CFTR is not spliced super effectively. There are a number of isoforms which are not full-length CFTR and do not produce functional CFTR. However, the superexon only has 1 splicing event. And so I think we're kind of compensating for a reduced mRNA stability with highly efficient splicing. Although, again, this is something we need to look more into. And data I didn't have time to show, overall, the superexon was spliced with high fidelity, and we weren't seeing very much of that bypass splicing at all. So moving forward, we wanted to shift our focus to superexons inserted in the more 5' end of CFTR intron 1. What I showed you in the last couple slides had a very 3' insertion site. And that's because we want to optimize constructs that would help more people with CF, including those with the del2-3 and del2-4 CF-causing mutations. And I want to thank Gary Cutting and his team for their work on CFTR sequences and variant determination, which allows us to really precisely understand where these mutations are occurring in people with CF. Now, even though they're called del2-3 and del2-4, both of these mutations actually include deletions that include about 6 kb of intron 1. So if we focused on the first 18-ish kb of CFTR intron 1, a superexon could benefit people with these mutations as well. However, as we think about continuing to optimize the superexon, both the insertion site and the mRNA stability, there are a lot of variables. Where to put it? 18 kb is a big region. What splice site to use? What sequence? Do we need a mini intron? Mini introns have been suggested to potentially help increase mRNA stability. And we also know that the polydenylation signal sequence can be really important. So in order to try and look at this in a more high throughput method, we developed a nanolook superexon so that we could evaluate this in a higher throughput way. So instead of the CFTR coding sequence, we now have the nanolook coding sequence and a peptide linker. And when this is inserted into CFTR intron 1, you get a funny little fusion protein that's CFTR exon 1, a peptide linker, and nanolook, but the nanolook is functional. Now, the nanolook readout is dependent upon a couple of things. It's dependent upon gene insertion frequency and fidelity, construct expression, so splicing and other factors. And because this is being driven off of the endogenous CFTR promoter, endogenous expression of CFTR. So we first wanted to look at a number of different combinations. So we looked at 2 different insertion sites, 2 different splice sites, the presence or absence of a mini intron in one location. There are a lot of variables here. and 5 different 3' UTR polyadenylation signal sequences. And importantly, we made every combination of each of those for 40 total constructs. We made the linear double-stranded DNA for these 40 constructs and delivered them along with conventional CRISPR components to 16HB14O- wild-type cells via nucleofection. Sorry, I'm pointing on the wrong screen. Via nucleofection. We normalized for cell number with Alamar Blue and measured luciferase with a nanoglue assay. And importantly, the data I'm going to show you on the next slide represents data from an experiment that was conducted 3 separate times across 3 weeks for 3 completely independent and biological replicates. And this is what the data looks like. I think upon first glance, you'll appreciate that there are many differences or major differences between the expression of these constructs. For example, it's pretty apparent right away that insertion site 2 and splice site 2 do not pair very well together. And there's a lot to unpack here, and I'll just show you one example. Shown in red is the same insertion site and the same splice site, same UTR, the absence or presence of a mini intron. In this case, the presence or absence of a mini intron really has no notable effect on the expression of the nanologue construct. However, in another case, same insertion site, different splice site, different UTR polyadenylation signal sequence, the mini intron has a really notable effect on the expression of the nanologue construct. Now, we also wanted to correlate this data with primary HBEs. Now, because undifferentiated primary HBEs do not express appreciable amounts of CFTR, we needed to differentiate these first. So the experimental outline is a little bit different and frankly more complicated. So we delivered the components to wild-type primary HBEs via nucleofection, allowed them to recover, and then differentiated them for 4 weeks at ALI and read nanoluciferase. Now, to start, we only started with 6 constructs here instead of 40 in the primary cells. And this is the data. On the y-axis is the normalized 16-HBE data. On the x-axis is the normalized primary HBE data. And while the correlation isn't perfect, we were excited to see that the constructs that didn't work well in the 16-HBEs also didn't work well in the primary cells. And the constructs that did work well in the 16-HBEs also did work pretty well in the primary cells. So we need to repeat this with additional constructs and further evaluate this correlation. But we were initially pleased to see that this appeared to be correlating pretty well into primary cells. So to summarize part 3, a nanologue superexon in CFTR intron 1 is functional, and this can give us a nice tool to evaluate genetic components of CFTR superexons. Genetic components all interact to affect CFTR expression. They're not modular. They must be evaluated independently because they interact. We're also currently in the process of determining the correlation between nanologue superexons and CFTR superexons, and obviously this will be critical for determining how far we want to push the nanologue superexon. So take-homes, if you take nothing else away from this talk, I hope that you'll take away that CFTR superexons can work well, but they need optimization. And we've developed some tools to help expedite that optimization. So future directions include more optimization, evaluation in primary cells and animal models, and that's where our collaboration with Craig and Ron comes in, and continued collaboration with several academic and industry partners to match optimized CFTR superexons with novel gene editing mechanisms and delivery modalities to get it done. So I had the privilege of speaking about this today, but this was really a team effort, and I want to acknowledge the many people involved in this work. I want to give a special thanks to KZ Yan, an extremely talented scientist on my team who has been spearheading the exon 2 through 27 superexon work in the 16-HBEs, as well as the nanologue superexon work. And he was assisted in the nanologue superexon by Caitlin Macadino. Kath Bucas and Elisa Bell have both made substantial contributions to all of our superexon work. And most recently, Elisa has been leading our work with superexons in primary cells. We have a fantastic next-generation sequencing and bioinformatics team: Mercy, Matt, Norm, Josh, Barbara, and Andre. The CFTR functional assays were from Yi Chen and Kevin Coote. I also want to give a special thanks to Cal Cotton and Martin Menz for their guidance and advice on this project. Also, we have great collaborators in Craig Hodges and Ron Conlon who are really pushing this to the next level in vivo models of CFTR. And then the 16HB14O- cells are originally from the late Dieter Grunert at UCSF. And with that, I'd be happy to take any questions.
Thank you very much, Hillary. We have one microphone that I can see here in the middle of the room. So maybe we'll start with you, Patrick.
Yeah, that was a lovely talk, Hillary. My question is, what plans have you got to look at the superexon in either other cell types or other tissue types, specifically to compare the level of expression as it changes? Like, you're talking about optimizing, and it's really cool to understand what's going on, but will you get the physiological levels? Or is that possibly something that you might see with Craig and Ron and the mouse models there to give you some handles on those questions?
Yeah, thanks, Patrick. That's a great question and something we think a lot about. I think that that's something that we're best set up to look at in the mouse model. If you were to look at, different cell types, 16HBs, KCO2s, there's a lot of differences there as well. I think if you're looking at an individual mouse and different tissues from that mouse, I think that's really the best way to compare expression across different tissues. And that's something that Craig and Ron are planning to do. And we're really interested to see how that shakes out because, we really have no idea what this will look like in the intestine or the pancreas. So we'll see.
That was great. I'm excited to see the matrix of possibilities grow. So the insertional effect paired with the mini intron is how they don't it's kind of fascinating. Did you look at alternative splicing with those different positions in the mini intron to see if that's a possible mechanism?
Yeah, so there are a lot of possibilities with the mini intron. In this case, we used one particular mini intron at one insertion site, and we were looking at just the presence and absence of that. And the readout here was really nanoluciferase. We had a lot of constructs. We haven't gone back and looked at the splicing of those, but that's definitely something that we could do. We're using this more of a screen and trying to understand which ones work well as opposed to trying to understand, frankly, why they're not working well. But it's certainly something we could go back and look at.
Well done, Hillary. Really exciting. Cells have ways of moving DNA in and out of specific locations. So if you get an insertion, it's not always going to be stable. If you consider the experiments long-term to take a look at how stable these insertions are and whether or not they're full-length insertions and how much heterogeneity you have. One thing I was thinking is, can you do single-cell DNA sequencing to start answering some of those questions, or do really deep sequencing to take a look at this question? You got the team to do it, but I think those are going to be key questions as to how often do things move around and how stable is in that location.
Yeah, great question, Gary. Actually, we have our long-term study ongoing right now. And so we're keeping them out for, a number of months to be determined. And actually, Kath just took a quick look, and I think it was 3 or 4 months out, we still see really robust CFTR expression. So that's all I can comment on at this point. But we'll all have to wait for that data. In terms of the sequence, in terms of the insertion, so we're doing this with conventional CRISPR-Cas9 based approaches, but we would imagine that in people, we might do this with a variety of other techniques. And so, we did see a couple clones with partial insertion, but that's due to the editing mechanism. And again, we're really focusing on the cargo optimization. And so I'm not too concerned at this point about partial insertion because this is almost certainly not how it would be done in reality. But we could follow up with sequencing in that. I mean, they're clonal lines, so it would be pretty easy to look at. Thanks, Gary.
It's fantastic work, Hillary. Thank you so much. And I love this idea of a superexon and everything. And my question really kind of answered it a little bit there. Given how many different SNPs there are in people with CF, an intronic sequence probably has even more. Is it feasible to just have one site to integrate, or are you eventually going to have multiple different of these sites? What's the thinking there?
Yeah, fantastic question. And that's something we think a lot about because we obviously want this to work for everyone or almost everyone. And actually, Shahram in 2 talks is going to be talking about genetic variability in CFTR and what we need to think about both in terms of targeting as well as off targets. So I don't have any specific data to think about to comment on that, but it's something we think a lot about. And I think, as a field, we're starting to coalesce around the idea we need to take that very seriously.
Hey, Hillary, great talk.
Thanks, Shahram.
So why the focus on an intron versus, for example, why can't you put it in the beginning of exon 2, right? Is it mostly because then you cover other variants like the exon 2-3 deletions? Because the reasoning would be that then you wouldn't have to compete with the native splicing regulation and things like that, right? And also, would you consider things differently? For example, if you have insertion mechanisms which have LTRs, and is that why you're thinking about insertion into an intron?
Yeah, so if there was a very clean insertion mechanism that could do a 4.5 kb insertion, I think we would seriously consider exonic insertion. But that doesn't, we don't really have that right now. And so I could spend the next 20 minutes talking about the different insertion mechanisms, but I won't. But, there's if you use something like a recombinase, you often have a scar left behind. So that's not going to be appropriate for an exon. There's a number of CRISPR-based approaches where you might still have a low level of indels or a low level of on-target editing that is not what you desire. And so with the current editing technologies, and this could change at any time, I don't think it's really feasible to do it in an exon in a person. Yeah, but that could change, and I would love for that to change.
Great. Thank you very much, Hillary, and thank you for the great Q&A. I'd like to introduce our next speaker, Jeremy Duffield, coming from Prime Medicine, and he'll be speaking on prime editing to correct genetic hotspots and prevalent mutations causing cystic fibrosis.
I guess we'll just, when I move it forward, then just click it. Okay. Well, it's great to have a wonderful audience on a Saturday. I thought everyone would have gone home, so incredible to see you all here. Thanks so much for the invitation. I spent nearly 6 years at Vertex. Don't know many of you quite so well, but know cystic fibrosis very well. And when we set up Prime Medicine, we founded the company in 2020 in the middle of COVID, based on David Liu's and Andrew Anzalone's new technology with prime editing. And one of the first things we thought about was, could we come up with a curative genetic approach for fixing mutations in cystic fibrosis. So I'm going to talk to you a little bit about that and about the company and where we're heading and how we think we could really help the community here. And I'm just going to be a little bit challenged with the technology. So I'm just going to give you a very brief, because I know you know this a lot, so I'll whiz through this, a brief overview of the challenge for fixing the problem for patients with cystic fibrosis. Give you a quick introduction to the prime editing technology and how it really is differentiated from other editing technologies. And then I'm going to talk to you a little bit about our liver platform. This is not a liver organization, but I want to show you what we've done in a very short space of time in the ability to correct pathological mutations causing liver diseases and metabolic diseases. And then I'm going to switch to the lung and talk about how we're building on that liver platform to develop editing technologies to fix causative mutations in the lung. And then finally talk through our approach for cystic fibrosis, which includes developing editors to correct genetic hotspots. And we're going to highlight just one of those mutations today, G542X. So I'll just be very quick here because I think many of you are so familiar, but this is a challenging space. A lot of mutations you just heard about on the left. The barrier of delivery to this tissue, which is naturally a barrier to organisms and pathogens. The challenge of getting to those basal cells if you want a permanent edit or a permanent correction. And then we have an additional challenge that, unlike some of the other tissues that we're working in, the translational models to give you really confidence that your drug candidate is actually going to be successful in humans, they're really hard. So these are some of the challenges. And then for all of the delivery technologies, they all have their pros and cons, and we're all grappling with those. So these are the challenges that we see, but we think a lot of progress has been made in how to overcome those. And this is a rather busy slide, but it gives you that potted history of how genetic therapies have been developed for cystic fibrosis, going back to the chastening times in the early 90s, where adenoviral vectors caused inflammation and didn't result in a notable expression of the delivered transgene through real progress with AAV systems. And I think we've just heard some incredibly exciting data in the last few months on AAVs effectively delivering a genetic cargo to the lung epithelium. But they still have their challenges, short-term episomal persistence, safety concerns still, and obviously distribution to the right cells in the lung remain challenges. And of course, the cargo size for an AAV is also a challenge for this large CFTR gene. Whizzing through the lentiviral vectors, which have not made nearly so much progress, we've had a lot of excitement in the last few years with the mRNA LNP approaches, firstly from Translate Bio, but now with clinical trials ongoing in the lung from Recode and from Moderna and Vertex, suggesting that we can use a non-viral system to deliver effectively to the lung epithelium. And that's to be proven, but I think we're all very excited about those possibilities. And then, with the advent of gene editing and now prime editing, we think building on all of these learnings and all of the capabilities that others have gone before is an opportunity for us to really help the patients with cystic fibrosis. And I'm just struggling a little bit with the moving of the slides. Seems to have stopped working. Okay, so here's something I want to talk to you briefly about. This is from a publication, but there are many such single-cell RNA-seq data sets that you can get access to. The key point here is that, unlike the pancreas, the CFTR gene is incredibly tightly regulated. And so if you think about developing a transgene and putting it into the lung, you're probably not going to really restore normal gene expression. And probably the reason that very few cells in the epithelium of the lung actually make CFTR is probably important. So this is a data set just showing you that only 6 to 7% of lung epithelium expresses any CFTR. Just shown here, of 16,000 lung epithelial cells analyzed by single-cell-seq, only 1,000 of them have any CFTR. Here they are shown on a phenotypic map. But the other really interesting thing, and it's been shown time and time again, is the number of transcripts in any epithelial cell in the lung is incredibly low. So why is that? That's probably really important to the function of CFTR in the normal lung. So if you imagine you saturate ciliated cells with thousands of copies, that may not be a great thing. This for us is really important because we really want to try and restore normal gene function. That's what prime editing offers the opportunity to do, and that's been one of our reasons to focus on. Can we fix the endogenous mutations back to wild type to help patients? So in a nutshell, this is prime editing. I'm not going to go through all of it for you. You can go to our website. We actually have a movie that lasts anywhere from 2 minutes to 5 minutes that shows you how prime editing works in great detail. It's incredibly elegant technology built on the shoulders of giants in some ways. The Cas enzyme has been heavily modified, fused to a reverse transcriptase enzyme. And the guide RNA has been incredibly heavily engineered. It's a little bit like a nuclease guide. It has a search domain that takes you to that unique place in the genome. But the technology is really, really heavily developed in what's called here simply a replace sequence. But this brings in the sequence that you want to put into the genome. The editor finds that place in the genome. It doesn't break the DNA. It just makes a nick, opens this flap. The flap then binds to the opposite end of the RNA guide through sequence complementarity, activates the reverse transcriptase that then straight into your genome writes in the normal sequence that you want to have there, and that's shown with blue here. There's a number of steps that are required to finalize this that are all highlighted in great detail on our website, but this results in, without any breakage of the DNA, a correction back to normal in your endogenous genome. Sorry, I'm just struggling here with the control. So this is a rather busy slide, but this is also available on our website. What prime editing can do is that it can correct any substitution back to the normal sequence. It can correct any small insertion or small deletion. And over the last few years, in combination with Prime Medicine and also David Liu's laboratory, we're now able to remove very large pieces of DNA, kilobases of DNA, very precisely without double-stranded breaks. And we're now even able to introduce, without double-stranded breaks, very precisely kilobase-sized pieces of DNA. And certainly the technique you've heard about the Superexon is something that we can combine prime editing with to introduce the Superexon. But I'm not going to talk about that today for the sake of time. The technology is highly programmable, and I'm going to touch on the fact that we can now edit larger and larger regions just with a single prime editor, so that we can actually correct many mutations with a single prime editor. I think one of the very promising things for the technology is so far we're really seeing very few or undetectable off-target events in the genome. And obviously for developing genetic therapies, a major concern is what do you do that you're not intending to do? And I'll show you a little bit of data, but so far for our lead 4 programs, we haven't found a single off-target event, which is just incredibly exciting. Obviously, this is permanent like other editing technologies, and the breadth of what we can do with this technology is incredibly broad. So we need your help because we got to stay focused to deliver. So this is just a high-level schematic on how we can do hotspot editing. I think you're all familiar with some of these exons. This is not the CFTR gene, but you can imagine 4 different mutations in exon 8 here. We can develop a single editor. That single editor has a corrective sequence that corrects all of these different mutations. So you can develop a single product here, correct all the mutations in a single region. We can do this effectively now for over 100 bases. It's actually been reduced to practice for 800 bases. The efficiency is not quite there, I think, but certainly for 100 bases, we can correct a whole region. So very exciting, particularly for CFTR, where you've got the spread of mutations, but they do indeed cluster in hotspots. And I'm just showing you one example from one of our retinal degeneration programs. So this is not cystic fibrosis again, but just to show you this gene Rho, or rhodopsin, is mutated. It's one of the commonest causes of retinal degeneration. We're developing prime editors to help these patients. And one cluster of 18 mutations is found at the C terminal of the rhodopsin gene. And we're just showing you here a heat map showing we have prime editors that fix, a single prime editor fixes with high efficiency all of these mutations are shown here. So really quite remarkable, and we can address 30 to 40% with a single editor here. And we're just showing you here in a humanized mouse retina that when we edit, we can correct all of those mutations back to normal with 70% efficiency in the humanized mouse retina. So very promising for those patients, but we think we can apply the same technological learning to cystic fibrosis. And I'm going to pivot now and just talk quickly about our liver programs, and just to help us think about how we can potentially move things forward for cystic fibrosis. So we've spent a lot of time engineering and developing a delivery system to the liver. It's a highly engineered lipid nanoparticle with a targeting ligand that specifically targets hepatocytes. And it brings in the prime editor as an mRNA that encodes the enzyme component and the guide RNAs that I just showed you. And this, from a regulatory point of view and from a commercialization point of view, and for a path to patients, is actually incredibly helpful because potentially we just swap out those guide sequences and the rest of the system remains the same. And from a manufacturing, controls, regulatory path, this makes it easy for us to, or relatively easy for us to, bring next programs forward. And certainly that's something we're very actively thinking about with regulators, is how do we bring forward multiple highly similar products without needing incredibly long and very expensive paths to satisfy the regulatory system. So just very high level schematic, we build these highly engineered mRNA, these guide RNAs. We formulate them in this LNP system that is targeted specifically to hepatocytes, and we deliver this by the single IV infusion, and it goes to the liver and it edits the genome. And I'm just showing you from one of our diseases that we're working on. It's a disease called glycogen storage disease type 1, where it's mutations in this G6PT gene. It's a disease of glycogen storage, so patients can't break down glycogen because the enzyme and the enzyme system is missing to break down glycogen. So when these patients are fasting overnight, they develop hyperglycemia and severe consequences. So this is a very prevalent mutation in this disease. It's a frameshift mutation. It's a loss of a C and a T in this very specific point in the gene. It leads to no protein production. Prime editing can very precisely put that C and T back in. And here are the data down below. So when we do this in a humanized mouse system here, you can see that we have a dose-responsive editing system that almost completely saturates correction. And in the liver, there's only 60% of the liver is hepatocytes, so we can't hit more than 60%. But you can see in a dose-responsive way, we essentially can correct that mutation back to normal. And then if you look in that region, 300 bases either side of that fix, almost undetectable other outcomes. So very different from CRISPR-Cas9, we're almost undetectable, and these are actually rare substitutions that we're finding. So it's an incredibly precise technology. And then on the right, I don't have time to go over all this today, but this is part of our off-target pipeline. And what you can see here is we look at potential candidate off-target sites that we've identified throughout the genome in patient cells. And when we measure them with deep sequencing, we can readily find the editing site that we've intended, but we haven't found a single off-target site anywhere else in the genome. And just very quickly, as you might expect, when we fix the mutation here, we correct the protein. So here's these mice that don't have the protein. Here are some different editors. Each one's got a different efficiency, and we're getting correction of the protein back to normal. Protein's found in the endoplasmic reticulum of the hepatocytes. And as we would predict, when we edit with a certain efficiency, the protein correction matches to that, as you would predict with a precise correction in the genome. And then we've taken this forward now into primates, and I just want to show this data because within a small amount of time, we've gone from concept to primates with that same editing system. Here, these are healthy primates. We're making a very similar point substitution in this case, in the same location, in the same gene in the primate, with a very, very similar editor that we think can help with regulatory interactions. And you can see that in these primates with a single infusion that's highly well tolerated, that we get high efficiency editing in different cohorts of animals here, 2 different cohorts. You can see we've achieved almost 50% editing in whole liver, which we estimate is over 80% of the hepatocytes are precisely edited. So incredibly exciting that this is working in primates. On the right, what you're seeing is unintended edits. Around 300 bases of the edit site, we don't find a single unintended edit here. So very, very precise technology. It's working in primates in the liver. So I think the question for us is, can we do the same in the lung? Is the same concept going to work in the lung? An LNP that's tuned to the lung epithelium, potentially with some sort of targeting system, could be delivered to the lung and can make these similar corrections. So that's the concept. I think we're all incredibly heartened by the groundbreaking work of others, the collaboration between Moderna and Vertex, establishing LNP delivery through local delivery, through the airway, potentially systemic delivery through a systemic infusion could be an alternative, and that's being worked on by Recode and the Seagwalk lab, although Recode's also developed this system as well. So we think people have made great progress with delivery to the lung epithelium. Of course, it's a new challenge. But among others, we've also been working on this. We've built internally an extensive lipid library. We have 17 chemists building lipids for us. We have a screening system where we can make hundreds of LNP formulations and take them straight in vivo and screen rapidly for highly effective lipid nanoparticle systems, and we can read out prime editing. And just to give you an example of that, here's one experiment where we were able to formulate 100 lipid nanoparticles, all different, all containing a prime editor. We're able to multiplex these. Here they are all made, and then they're injected intravenously into a mouse, and then we can purify out basal cells from the lung epithelium here, and we can identify which of those LNPs, because they're all different, which of them was very effective. And you can see here just one example, one of those LNP systems, edited 40% of the basal cells. From the sorted basal cells. So early days for us, and I think we're certainly behind others, and we're certainly interested in collaborating to try and get the technology to patients quickly. But certainly we have, I think, initial proof of concept that we can develop nanoparticle systems that can deliver this large complex cargo to basal cells. Just to touch on this very quickly, we're as a industrialized company, to make prime editors is difficult. It's not like a CRISPR guide. We actually have hundreds to thousands of choices of guide RNAs, unlike 5 to 10 that you might have for a CRISPR screen. And so in order for us to find those highly active guides, we've had to build industrialized high throughput screening systems. And just giving you a high level schematic here. We've built this at an industrial scale. We're now able to go from idea to hits that are highly active within as little as 3 months. Here's just some of the capabilities. We're able to screen now 22,000 samples a week, where our next-gen sequencing can go through 15,000 samples a week. And this really is enabling us to industrialize this technology. And then on the next slide, just quickly, we've built actually, I'm not going to show that today, but we've built a machine learning algorithm system so that all the guides that come out with the editing data, we're able to screen out now without actually having to do screening. We're able to screen out guides that don't work very well. So we've cut our total amount of screening from thousands now to about several hundred through using a machine learning system that's built into our software. And then I think a really other important thing, we've built a company to make medicines. So we are just touching on this, just one example. But we've built comprehensive synthetic guide RNA production. These long guides are really, really hard to make. We've broken new ground here. So these long guides at high purity are definitely hard to make. I think we're now leaders in the world in this. We have incredible mRNA technology, and we're making LNPs that can work through to phase 1, and we're just showing you those data from the liver. So this is just one example where we're making these very long guide RNAs. Some of them are 150 to 200 nucleotides long. We're making them with these synthesizers, and we've developed systems to now make very high purity guides that are well above the purity that's required for regulatory interactions. And so again, we're doing this at scale. We're making them for primates. We can make them for humans now. So we think we're ready to go if we get the delivery system right. So just in the last few minutes, I'm just going to flip and turn to you, talk about the approach for cystic fibrosis. I've sort of alluded to it. But, we think we can develop a small number of prime editors that can correct 7 or 8 hotspots in the gene. And by doing that, we may be able to address the vast majority of patients. Of course, bringing forward 7 or 8 different products to regulators has its challenges. But we think this is at least one way forward. And of course, gives us the advantage that we're correcting the gene back to normal, just like the gene you and I have. And so this is just a map taken from Molinsky's paper in 2018, just showing you the allele frequency of all the different mutations. And indeed, there are lots of mutations. But of course, many patients are compound heterozygotes. And you only have to fix one copy to have a curative approach for patients. And so our calculations are that if we can actually develop these 8 hotspot editors here, actually we cover the vast majority of patients. And there may, of course, then be relatively straightforward ways to capture the other patients who still don't have a therapy. So we've focused on these nonsense and frameshift mutations that are high unmet need, as well as other mutations that are not responsive to current therapies. And we're just going to show you an example of this, but we have multiple hotspot editors that are in various stages of development at the moment. So here we've used our screening systems to find editors that precisely correct this prevalent G542X mutation. We're showing you here that in patient cell system that when we first started, it was very low efficiency, as you might see, but with optimizations, very quickly, we're able to improve our editors so that individual editors are hitting 70% precise correction with no mistakes from a single administration. So very high efficiency out of our screens and initial optimizations. But of course, that is the first stage. So, oops, I'm sorry. How do I go backwards? This isn't working now. Sorry. There we go. So we've developed internally a human intestinal organoid system. So we have G542X homozygous null stem cells that we can differentiate into organoids. And what we're showing you here is that this swelling assay, I think, is very familiar to many of you. It's an intestinal organoid swelling assay where when the CFTR protein works normally, the organoids swell in response to forskolin. But when the CFTR is mutant, they don't swell. And so what we're showing you here is that we can edit directly these epithelial cells using an RNA LNP type approach. And you can see that healthy organoids swell normally. The patient organoids don't swell. When we edit these organoids with a prime editor, now they swell normally. And as you might expect, Trikafta, which doesn't address this mutation, doesn't work. I'm just showing you here on the left that, actually, in the total number of cells in these assays, we don't need to edit that many cells to get a complete correction of swelling. So also very exciting that we, have a highly efficient system. And then, like others, we've been working closely with collaborators, and I think Craig Hodges' group, who are here today, this is data that we've done collaboratively with them. They've developed a mouse that has a human exon 12 that has the G542X mutation, and the groups there got incredibly adept at taking intestinal stem cells, and we've shared our editors with them, and you can see that they edit directly the stem cells, and then they differentiate them into organoids. So this is a little bit different experiment, and of course this is a humanized mouse, but you can see from our editors here, we have editors that are hitting 60% precise editing with no mistakes in the stem cells, and indeed they are correcting the swelling of these organoids in a totally independent set of hands. And here's just the swelling of those organoids shown at the bottom. So we think for this mutation, we're making great progress, but what I'm not showing you today is we're making great progress with additional hotspot editors as well. And so we think that this is a viable approach forward. And then I think really the last slide is just to say, of course, that's intestinal organoids, but of course this is working in human bronchial epithelial cells, and these are bronchial epithelial cells, again, from a collaborator for G542X. And just to show you that our tool LMPs, these are early days, but we're already getting, 30% editing from this one and 20 from this one. Obviously, we can improve these further, but there's a possibility of redosing as well. And so we think we're making great progress to next step, taking these into humanized animal model systems. So it's a bit of a whirlwind, and happy to take any questions, but I'll just mention a couple of things again, just to remind you, we're very excited about this technology. It's working incredibly well. Our first clinical trials, we hope, are going to be starting early next year. Fingers crossed, but things are going well. I showed you liver data in primates showing that the technology does what we thought it would do 3 years ago in primates in vivo, that we can deliver it effectively and safely using an RNA LNP system, a little bit like the COVID vaccines have been developed. And I've talked to you about one approach, complementary to the Superexon, and I certainly think prime editing can help with the Superexon approach as well. But we think one approach is to correct hotspots in the gene, and I think we have the capability to do that now. And then obviously, definitely looking for help with this. LMPs that can get the prime editing system to basal cells for humans, for patients, is a big challenge. We're certainly making progress there, but we're open to talking with collaborators to find out the best path forward. So thanks very much.
Great. Great. Thank you very much. That was a fantastic talk. Maybe I can start with the first question, and that is, how do you see the pathway forward before IND, let's say for CF? And I think the first question is what preclinical demonstrations do you believe you need, either in CF animal models or simply a non-CF, non-human primate? And secondly is, once you've gone through the pathway, let's say for targeting one of the prevalent hotspots, do you see that there's sort of an ease then moving and getting approval for second and third and fourth hotspots?
So lots of great questions there. So, we have a path through R&D, which involves patient cells, patient model systems, humanized mice, because the prime editor is so specific for the human sequence, it doesn't work even against the primate sequence. And then the question is large animal, because obviously scaling from small animals to large animal, the biodistribution, the tolerance, all these things are critical. So we have developed a surrogate editor system, which I showed you for the liver disease, where the editor is almost identical to the human editor. It hits the same spot. The drug looks almost identical. we think that may be an approach, even for CF, going into healthy primates and making an edit that looks almost identical. That's one path, but of course, there's still a question around the abnormal lung environment, how that responds. And I think one thing we're thinking about is, there are great primate ferret models around. And so there's a question about whether we'd want an additional large animal data set as well. And then the question around the regulatory path, which is, I think we're all very actively talking with regulators at the moment, in various capacities. And I think there's a great willingness to find a solution here. But of course, regulators are here to make sure what we're doing is safe and we're not going to harm people. And so my hunch is there's going to be a path where the onus is on us to demonstrate safety and efficacy and then comparability. So I think there's a path, but it's not going to be, tomorrow it's all going to magically come into place. But, we may imagine that some of the early programs that come forward with these kind of technology may lay the groundwork for the follow-on programs.
Good, thank you. Questions?
Just a quick question. Do you all have any insight into the mechanism by which LNP makes it from the bloodstream, basically through the endothelial layer, and then into basal stem cells when you give it systemically?
That's a great question. I think we don't fully know the answer to that. We know that that they obviously biodistribute. Now, do they go through endothelial cells or do they go around endothelial cells? These are great questions. We haven't fully resolved that.
Have you looked at inflammation to see if the junctions actually are opening up at all?
We have not looked at that. We're very sort of we've been very much looking at the endpoint, which is editing in basal cells. I don't think we're fixed on the IV approach. The greatest progress has been with going straight to the lung. But I think the question is, if we really want to do this once or twice only, we got to hit those basal cells. So that's going to be a key question for whichever approach we take through the airway or through the bloodstream.
Really nice. Your liver data there in the non-human primates, you made a silent change in a leucine codon, and you get about 80% editing. But that gives you a really nice system to look at the long-term effects of editing. You're not seeing off targets, but you see over time, does it stick at 80%, or is there selective advantage for or against the edited cells?
Yeah,
really cool.
So we have long-term studies ongoing, so we'll be able to let you know about that. The data we've seen from elsewhere from base editing is that they've shown editing data in the liver out now to, I think, a year and a half, and it's been completely constant and stable. So that's on us to prove that. But I think there's a precedence that single edit can be stable in the liver.
Hi, great work. This is May Lewis from University of Texas at Austin. So I'm actually just getting started trying to design like a prime editing systems for cystic fibrosis. And so I was just kind of curious, like in your screens, I know that David Liu has like a lot of iterations for how to make these PEG RNAs better.
Yeah.
So I was wondering, is the design of like the primer binding site and the RT or the 3' motif like more important than the LNP, or is it the other way around? Can you comment on that?
Improvements everywhere lead to great outcomes. I'll take that offline with you. It's a long story, but there's thousands of steps to get to where you want to be, and so you have to improve every single piece. And so, but we can take more of that offline. Happy to talk to you at the end.
Thank you.
Hi, I am a parent of a 15-year-old with CF who's not on a modulator, and I do not have a science background, but I'm trying to figure out if R560T would be in that hotspot. She has an X mutation in R560T just because.
Probably 5602 is probably not.
T.
No.
Okay.
But I'd be happy to talk to you about where your daughter's mutation is. We could perhaps chat at the end.
And does this impact X mutations?
Does this impact
X mutations?
So this was an X mutation, and we're certainly targeting X mutations. We think that's where the highest unmet need is right now.
Okay, so all X mutations, or most?
So
Okay.
we've so far gone after X mutations that are in spots where there are lots of patients, but we certainly are. I'd be happy to talk to you at the end about how we can get there for individuals as well.
Okay, thank you.
Fantastic. Let's thank Jeremy again. Our next speaker is going to be Sharam Vidyanathan, who's going to be talking about genetic variability in CFTR and things we need to consider around that.
All right, good morning, everybody. First off, I'd like to thank Dr. Valley and Dr. Davis for inviting me to present here. So today I'll be talking about the diversity in CFTR variants and considerations for CFTR gene editing strategies. So let's see. There we go. Okay, so I think the first two lines here, everybody's familiar with that, right? Like, so we know that there are about 70 to 100,000 people with CF worldwide. And these are mostly documented, or these people have mostly been documented to be living in Europe and North America. And over 90% of this documented population can be treated using modulators. Now, we also know that CF affects people of non-European ancestries. And this is sort of data that most people are familiar with, right? Like it's more common in people of European ancestry, and then it really goes down when you start looking at people of other ancestries. And I was really happy with that until 5 years ago when I was having a conversation with somebody at Stanford here, and I was still a postdoc there then. And so I got to learn about several South Asian people being treated at Stanford. And so this led me to collaborate with Zach Sellers, and we got data from the registries in the UK and Canada. And we specifically went to these registries because they classify South Asians separately from people originating in other parts of Asia. And to our surprise, what we found was that both in the Canadian registry, UK registry, and the registry at Stanford, about 60 to 90% of Asian people with CF were from South Asia compared to other parts of Asia, right? And when we used this, the UK data set to then try to predict the prevalence, it ended up being substantially higher than the numbers that you see being thrown around online, right? This is all good, and if you've seen my talk, this is really old news. So then what we've been asking this question since I moved to Nationwide last year is, can we confirm this with a larger data set? And so Lily Barr was one of the students that was working on our previous work, and she really wanted to continue looking into this a little bit more. So we actually went to Nomad, which aggregates genetic information. And so here we have information from over 100,000 different people. And so this data set is a little different from the previous data set, right? So this is data that comes from the general population, not from people with CF. And so we had to go in, and they do actually a pretty decent job of curating pathogenic versus non-pathogenic variants, but we still had to go, and we did find some sort of inaccuracies in terms of variants that were considered non-pathogenic or unknown significance, right? And so we essentially used Hardy-Weinberg equilibrium to then calculate out the prevalence in the Asian population first. And so what you can see here is, and we have good representation, so we have about 15,000 exomes from South Asians and about 10,000 exomes from East Asians, right? And you can see that this pretty much recapitulates what we published before, that if you look at South Asia, there's a significantly higher prevalence of CF at birth compared to East Asia, right? And then, of course, then we wanted to validate that this was not just a fluke and you could actually calculate out the prevalences for all the other groups, right? And so we did that using the Nomad data. And this is basically recapitulating what we already sort of know, but maybe not numerically, right? So what we see in the Nomad data set is, again, it's most common in Europeans, as we all know, right? And then it's actually not that uncommon when you look at people of Hispanic or African or South Asian ancestries, and really drops when you go into the East Asian part of the world, right? And so as we were thinking about this, there was this other paper that came out, Tabor et al., and this is from Myriad Genetics, where they do carrier screening. And if you really go into their supplementary data set and look at that table, they have actually carrier frequencies for 150 different disorders. And so we were able to use that to then calculate the prevalence for CF in different populations. And what we see here is that it really nicely correlates with the Nomad data set. You can notice that the Tabor et al. Estimates are always a little bit higher. And this just has to do with the fact that Nomad doesn't have good representation of intronic regions and probably doesn't have that good representation of large deletions and things like that, right? But what you see again here is that the prevalence really drops off when you go into East Asia. But when you go look at all the other parts of the world, there is actually a not so insignificant prevalence of CF. And of course, this is often where conversations stop, right? So how do we translate this into actual people living with CF, right? So what we started doing then was looking at countries by birth, right? So this is a plot of the top 10 countries by birth in 2022, and this is just based off UN data. So the first surprising thing is, well, it's not really a surprise. India produces a lot of people, right? And then if you look at the other 9 places, most of these places are either in Asia or Africa, and US seems to be like one of the few outliers here, right? And then you can actually try to predict the number of people being born with CF on a yearly basis. And what you see here is that when you compare a country like India versus a country like the US, right, the number of people being born with CF in both of these countries are actually pretty similar, right? I was really surprised by this. And then when you start looking at even countries like Nigeria, you're starting to think about not an insignificant number of people being born with CF. And of course, I wanted to sort of give some context, right? So we started comparing this with the prevalence of hemoglobinopathies, and we just used the carrier frequencies that were reported in Tabor et al., right? And so in some countries like India, they really recognize that hemoglobinopathies are a significant problem for them. But then I don't think they quite appreciate that the number of people being affected by CF might be comparable. And there were a couple of great talks earlier here by Dr. Varkey and Dr. Paul earlier in the conference, and they're actually quantifying the prevalence of CF in India, whereas this is all data coming from NOMAD and mostly from people living outside of many of these places listed here, right? So that was just to motivate us to sort of think about why we continue to need new therapies for CF around the world. Of course, the question is, can we just all treat them using modulators, right? So we've already just seen that there's just an incredible number of people living all across the world where we haven't characterized CF. We also know that work both that was done when I was at Stanford and work that comes from Dr. McGarry at UCSF is that when you look at these other populations, the number of people that don't respond to modulators is actually significantly higher than what you see in people of European ancestry, right? And so when you combine these facts, I think we need to start thinking about a larger CF population than we're accustomed to, and maybe a significantly higher fraction of them may need access to a new therapy, which then takes us to thinking about what are the variants that dominate this global population, right? And so here is a plot of delta 508 and the prevalence of delta 508 as it pertains to different populations. And you can really nicely see here that it drops as you go into different populations. It's really lowest when you go into the Asian population. And there's really no one variant that dominates many of these populations. And this is just an example from the South Asian population, just mostly because most of my work focused there. And you can see that there's a high prevalence of many nonsense variants and splicing variants. And so how do we think about correcting these variants, right? And so we've already seen many of these sort of strategies. I just want to remind people, right? So there's gene insertion using homologous recombination, where you use Cas9 or any other nuclease to induce a break, and then you can make a homology-based repair template. But of course, the disadvantage here is that it needs cell division to work, right? You can use base editing. And so base editing is just a nickase version of Cas9 fused with a deaminase, right? You have efficient gene correction, but then you have to go one mutation at a time. And then we just heard about prime editing, and it doesn't require cell division, and you can replace exons, right? So how do we think about all of these different sort of approaches to then think about how we can help people affected by different sort of variants, right? So the first one is to go mutation by mutation, right? And so this was my paper from when I was a postdoc. And so here our goal was to just use HR to correct delta 508. And we were able to get somewhere between 30 to 60% correction of delta 508, both in homozygous samples and compound heterozygous samples. And what you can see here is that by the time you get to about 20 to 30% correction, you can actually start seeing meaningful restoration of CFTR function. That's about the 10 to 20% threshold that people commonly attribute for making a therapeutic difference, right? So that's sort of our goal when you're trying to correct mutations individually. And subsequently, several papers have come out showing that you can use base editing or prime editing to then reach these levels of gene correction in different cell systems, right? So, but then the question is, how do you prioritize all of these target variants? I think the previous talk also sort of went into this in some senses, right? So Delta 508 is obviously makes sense. It sort of affects most people. And then some of these other variants we've seen throughout the course of this conference, right? And these have been really well characterized and known for a long time. And even some of these, right? Like, for example, this splicing variant is actually really, really common when you look at some of the Asian populations, right? And then as we sort of learn more and more and more about genetic variation, you can start to see that there's actually incredible diversity in CFTR variants. So the decision that we need to make is, do we go after each variant separately, or can we replace segments of CFTR like we sort of discussed with Dr. Bali's talk earlier, right? And so one strategy that came from Dr. Davis's lab was the idea of putting in a partial superexon in intron 8. And so you do get meaningful levels of restoration of CFTR function with a strategy like this, right? But then the question is, how does this affect people who are affected by variants in exon 1 through 8? And this is really only a problem if there are enough individuals who have biallelic variants in exons 1 through 8, right? It's really not a problem if most people are compound heterozygous for these variants. And so I went into CFTR2 and started looking at the percent of alleles that might be affected, that might occur before intron 8 that have mutations, right? And so about 5% of alleles in CFTR2 have variants before intron 8. But of course, I don't have access to sort of all of the other populations in the registry data. So I went into the data set that I knew, right? So this was the UK registry data set, and I mostly just looked at the South Asian population. And you can see here that about 10% of people with South Asian ancestry have biallelic variants that occur before exon 8, right? So here the exons are in parentheses. And so if you look at all the variants before intron 8, you can see this would add up to somewhere close to 10%, right? So then that takes us to this question of, can we replace the full-length CFTR cDNA? And so, of course, this is a challenge because of the size of the gene, and it doesn't really fit well into the packaging capacity of AAV. Even if you use the strategy that Dr. Bali suggested, which is to skip exon 1 and go into exon 2 or intron 1, and so you do 2 through 27, you still don't have space, right? So what we did, one version of our solution was that we were trying to put things in exon 1, but we split the gene into 2. And so we put in the first half using a first AAV, and we had a guide sequence in the end. And then we put in the second half of the CFTR cDNA, and then we put in a selection marker. So here is truncated CD19. So this is a B cell protein, but we took out all of the signaling domains so that you can enrich for these cells after you've edited them, right? And so here you have, as you might expect, this is a complicated sort of a strategy. So you have a low efficiency of correction. And so you get about 3 to 5% corrected cells. But because you have the CD19 cassette, you can then enrich for these corrected cells. And so once you enrich for the corrected cells, so here we have populations of cells that are somewhere between 60 to 90% corrected. And we tried several different variants, including many nonsense variants. And you can see that once you differentiate this enriched population of corrected cells, you can get robust restoration of CFTR function somewhere between 50 to 150% relative to what you would see in non-CF controls, right? So this is great, and it sort of validates that you can restore CFTR function if you put the entire gene in exon 1. Of course, the question is, can we improve this gene insertion further to avoid the need for enrichment, because that would make this so much more translatable, right? And so as I was finishing up at Stanford, there was this discovery that you can use small molecules to then inhibit non-homologous end joining, which competes with homologous recombination. So we tested this in the context of correcting delta 508. So you can see that without PK inhibitor, you can get about 20 to 30% correction. And then when you add in the PKCS inhibitor, this shoots up quite significantly to about 80 to 100%. So when I started my lab, so Jake has a poster, I think 260, and so you can still go see it if he has not taken it down. But and so you can see here that when we tested the same approach for the cDNA insertion, right, you can, when you add in the DNA PKCS inhibitor, you do see a substantial increase in the efficiency of cDNA insertion by about twofold. Some of these samples were edited to the point where you can think that you don't need any more enrichment, but a majority of the samples still require enrichment. So there's still more work to do in the area of improving cDNA insertion, whether you do it in exon 1 or intron 1 or any other place within the gene. But then, of course, the question is, how do we do this in vivo, right? And so all of this work was done in the context of a cell therapy. And so the idea here was to target people with CF sinus disease. And this was a collaboration with Dr. Nayak and Dr. Bravo, and they're in the audience. And so we were targeting people with CF sinus disease who are not responsive to modulators. And the idea is that if you have severe CF sinus disease, you would go into your ENT physician, and then they would debride your sinus, send us the tissue. We would edit them, put them on some sort of a biomaterial, and then transplant them back. And we just had a pre-IND meeting with the FDA last December. And so we're actively sort of planning to figure out how to sort of move this further. And Dr. Bravo had a really nice talk yesterday in one of the sessions, and she's in the audience, so you can chat with her a little bit more if you're interested. So here we actually screened several scaffolds to hold the cells in place when you put them back into the sinuses. And so we found fibrinogen as our lead candidate because it's a human protein that's not immunogenic. And then we looked at, Dr. Bravo looked at several different injury mechanisms. And so we want to go with mechanical injury for the human application, but then for the proof of concept studies, we used polydocanol. So we treated these mice with polydocanol to sort of make space for the exogenous cells. And then we put our gene-modified cells into the mice after encapsulation in fibrinogen. And you can see really robust engraftment of these cells for several months. And then when you stain the cells for both human EpCAM, you can see that there's human cells surrounded by mouse cells, showing that our human cells did engraft into the mouse epithelium. And they do retain expression of that truncated CD19 marker, which marks the cells that were corrected for CFTR. Right. And so this leads us to this question, right? Since we're talking about variants in this session, right? Can variants in exon 1 affect cDNA insertion? And this was, here is a list of, I think it's a partial list. I don't think it's a comprehensive list, but there's several variants in exon 1 that line up with the target single guide RNA sequence here, right? And so if you have people with CF who are affected by these variants, then inserting into exon 1 is maybe not going to be as efficient. Only some of these are pathogenic variants, right? And some of these are actually non-pathogenic variants. And so you might have an individual with CF where we may not actually appreciate the fact that they might have some of these variants in their genome, and that might be contributing to a lower level of editing efficiency. Now, these variants are incredibly rare. They're not super common, but I don't know if that's been thoroughly characterized in terms of whether one of these non-pathogenic variants is somehow linked to a pathogenic variant, right? So there's definitely more work to be done. These sort of variants still account for only 0.07% of alleles in CFTR2. And we don't know if most of these people with these variants are heterozygous for these variants. And so the take-home message is still that you can still insert the cDNA into exon 1 and help over 99% of people with CF, right? And of course, how do we think about genetic variation impacting off-target editing? And so there was a really nice paper from Dr. Luca Pinello's lab last year where they were looking at genetic variation across the genome and how that might change your off-target propensity, right? And so here they showed that when they picked a guide that was targeting an enhancer that's associated with sickle cell disease that's actually used in the product that's just going through the FDA review. And they showed that there's a variant in an off-target site where a low fraction of people actually have a new PAM site that brings online an off-target variant that was previously uncharacterized. And so this variant is present in about 5% of African Americans. Right. But they also really nicely showed that if you use a high fidelity variant of Cas9, you can pretty much completely abrogate this off-target activity, right? So here you have the reference allele and then the alternative allele. And then in the off-target site, when you edit using wild-type Cas9, this alternative allele is edited at a frequency of about 10%. But if you switch to the high-fidelity variant of Cas9, you can actually completely limit this, and this goes to somewhere below 1%, which is very manageable, right? So as we think about some of these other editing systems, right, maybe we need to think about switching some of these to the high-fidelity variant of Cas9 so that we can improve our targeting efficiency, right? And so the question is, has the use of high-fidelity Cas9 shown to be reducing off-target activity in the CFTR locus? focus. Of course, it has been. So we had used the high fidelity variant of Cas9 with all our exon 1 insertion work. And so you can see the comparison here. So there's 50 off-target sites that we evaluated for our guide RNA across the genome. And you can see that there's one guide site where there's significant off-target activity. And then when we switched to the high fidelity variant of Cas9, you saw about a 50-fold reduction in this off-target activity with this specific guide RNA, right? So with that, the summary is that there's definitely a need for alternative therapies for CF, especially once you consider the global population. And there are many ways of restoring CFTR. I think that's the take-home message from this session, right? You can go one variant at a time, partial superexon, complete superexon, right? And they all restore CFTR expression, so that function. So that's the good news. And then as we improve our characterization of both CFTR variants and genetic variation across the world, we would sort of improve our sort of ability to then go and treat 100% of people with CF, which I think is our ultimate goal. And with that, I'd like to thank my current group and then my mentor, Dr. Matt Porteous, and the Stanford team, and then all sources of support. And I'm open to questions.
I have a question, Shram, while people come up to the mic. So we talked a little bit about modulator eligible versus ineligible, and the many rare variants that are found outside of the U.S. How confident are we that those are modulator eligible or ineligible, or did you look at whether or not those have been evaluated?
Yeah, and this is a great question, and I think even as we were writing the paper, one of the more common variants that was previously ineligible became ineligible. And so that's actually a moving target. And you're absolutely right on that. And there's probably more work that needs to be done to characterize whether some of these variants are eligible for modulator.
Hi, Sarah. Really nice talk. I just wanted to say, you highlighting the fact that off-target mutations or off-target variants in different patients that can create new binding sites or off-target effects, or, we're still trying to optimize everything. And Hillary, I think I saw a poster by, the CFFT group of trying to, figure out what can we target most efficiently or where can we target, in different cell types that's going to work the most. But, we're all working with, established cell lines or, primary cell, we're not going to really know until we start genotype. Once we get to the point where we can actually do this with patients, unless we're, full genotyping, and we have to just, be aware that, one place that we're hitting to try to make these changes it might not always work for everybody because of, their complex genetics with just things that we don't look at all the time. So just highlighting that, I just want to say that was really nice.
Thanks, Jenny.
I think for the sake of time, if there are no other questions, we'll move on to our final speaker. Thank you so much, Shram.
Thank you all.
Our final speaker for the session is Phil Santangelo. So without further ado, Phil.
Thanks. Just see if that. Okay, so which one do you use for the? Mine.
Mouse has the little red thing, but if you move it over here,
Oh, then
this
it'll.
is what's actually on the screen.
Got it.
Try and move that more, but
It's okay. It's fine. No worries. First, I wanted to thank the committee for inviting me to give this talk today. This is going to be a little bit different because we're really going to be talking about delivery, not the emphasis on cargo. And I will admit that my lab actually doesn't really work on CFTR at all, or CF, but basically we came to this place really working on infectious disease. And so we were very interested we work on RSV, flu, COVID, other respiratory pathogens. And so it became quite critical for us to be able to develop vehicles for delivering mRNA to the lung. I should mention my lab also works on mRNA therapeutics in general. And so we had to find out of necessity a good way to deliver mRNA to the lung. So hopefully I'll give you a little more information about what we've been doing and kind of where we're going. Let's see if I can do this. Okay. And I did want to disclose a relationship with Tether Therapeutics, who will be taking some of these vehicles and moving them into the clinic, I hope, in the near future. So where are we now with delivery of mRNA to the lung? Some of the previous speakers talked a little bit about Translate Bio. They were I'd say one of the first companies to get an aerosolized or nebulized LNP mRNA combination into the clinic. Unfortunately, their results weren't quite what they hoped that they would be. Obviously, other folks, Moderna and Vertex, have an LNP formulation that they've moved into the clinic. So there's certainly been some movement, Recode also. But if you look at the literature in general, even with some of the newest papers, one coming from Dan Anderson's group in Nature Biotech recently showing some editing in a mouse, they only used intratracheal administration. And I want to keep folks in mind that intratracheal administration is great. It's a good way to show some proofs of concept, but you're not going to be able to do that in a person. And so you're going to have to nebulize. And there's a lot of limitations to nebulization that need to be taken into account. So even though this was certainly an interesting demonstration, I still think there's obviously some work to be done. There was some other work from the Saltzman group using a polymer formulation, and that's kind of where I'm leading, is to polymers. They also used IT administration, and they used it to vaccinate. And I think one thing I want to keep in your heads when you think about any of these vehicles is, what are they actually doing in the lung, not just in addition to delivering their cargo. And I think one question I had after looking at the Saltzman paper was, if you're using it as a vaccine and you're using a very clean mRNA, meaning it is modified with 1-methyl-pseudo-U, it's been hopefully purified very well. My question is, why are you getting an adjuvant effect? Because if you're generating if it's a vaccine and you're generating immune responses to that protein that you made, what's driving that? And so I would be asking about inflammation and adjuvanting from your own vehicle, because I think in CF it's going to be really quite critical that we deliver the cargo, but that we don't do any additional harm. We don't want to drive any inflammation. We don't want to drive those characteristics that would be, I guess I would say, fanning the flames a bit, in terms of we don't want to make things worse. So I just want to keep that in mind. So when we looked at cargo, basically delivery vehicles, we actually started with LNPs and with polymers. We were doing both. And from a polymer perspective, we looked at actually one of Dan Anderson's other papers, but on a PBAE-based vehicle. And we started actually with that. Let's see if I can hit the right buttons. We also explored LNPs, and that was work that we did predominantly with James Dahlman's group. And in the end, probably the most important thing for me to mention is that here are the things that basically why we're working on polymers right now as opposed to LNPs. One is lower expression. Honestly, so far with the LNPs that we've been able to compare with, we're seeing basically higher expression from our polymers. You'll see more of that in a minute. One other issue is when you're talking about your clinical development, I also, in the literature, often see mice only, and mice are great. We use them all the time, but the problem is that you need to move beyond mice. And so one thing that you'll see, hopefully I'm going to discuss a little bit, is that we got out of mice, at least initially, as fast as possible. And the reason is we wanted to see if the polymers would actually fail. So we moved from mouse to a hamster. Why we're using hamsters? Mostly for COVID work, to be perfectly honest. But just the same, different species. We wanted to see, would it fail? Then we moved on to ferrets, and a whole zoo, basically, is the way it's been described. And so, and honestly, it's kind of getting there in terms of the zoo. But the point was just that we wanted to see, A, if it would fail, if we could deliver into these larger species, would we see difference? So I'll get into some of that. But the point is, I think it's really important to understand if your delivery vehicle, your formulation is species agnostic. I think that's an important one. The other one is really, I think folks need to definitely concentrate on, is this issue of inflammation. So are you modulating gene expression in these cells in a negative fashion by delivery? And then the last one, and this may seem like an odd one really, is when you're talking about nebulizers, they're a whole beast by themselves. And there's different classes of them. There's vibrating mesh nebulizers that have been used in the clinic and jet nebulizers that have been used in the clinic. We personally think that jets might be advantageous. The reason being is you can get more, you can deliver your dose in a shorter period of time. I don't think anyone wants to be on a nebulizer forever. And so the reality is the lowest period, the least period of time that you're actually nebulizing to a patient would be helpful. The point being is that we wanted to make sure our vehicles were compatible with both types of nebulizer. And that's something that we've done. Now let's see if I can still change this slide correctly. Okay, so how do we screen in the first place? How do we develop these polymers in the first place? Well, really, it's a combination of chemistry, developing new tools, and in vivo screening. So we screen all of our particles in vivo, and we'll talk about how we do that. First thing we needed, though, is better reporter RNAs. Most of the classical ones, I would say, had some disadvantages. So we ended up with a GPI-anchored nanoluciferase reporter. So we use Nanoluc a lot. I was glad to hear it in some of the previous talks because we're big fans. Why do we GPI anchor it? I'll talk about it in just a second. In addition, we had to develop a screening platform that had low dead volume because most of the tower-based systems for nebulization use a lot of material. And that's fine when you're testing your lead candidate, but when you have to test 200 or so vehicles, that's very, very difficult. So we came up with our own inexpensive, low dead volume screening platform that worked with mice and rats and hamsters and ferrets, and we can use FDA-approved nebulizers in that process. And we thought that was important in order to move forward. Okay, so really it's this combination of all of the parts and pieces. Why do we use these GPI-anchored reporters? And we make lots of them. And the reason is that they're very, very sensitive. And if they're GPI-anchored, the GPI anchor itself, that sequence, causes the protein to be trafficked to the apical side of the cell, and it hangs out there for a while. So it accumulates, and it allows us to use very, very little RNA when we're screening. So I just want to keep that in mind. This is part of our screening approach. We also do work with Cas13, a different CRISPR protein. We've made Nanoluc fusion with it, with Cas9, with other proteins, and we actually use beta-gal for staining, which I know seems kind of like an odd one, but we have good antibodies to it, and so we don't even use the beta-gal enzyme activity. We just literally stain for it, but I'll show you some pictures of that too. But overall, we use this group of reporters. We use other reporters too. I didn't want to list them all, and we'll make reporters for certain problems. So a CFTR, say, Nanoluc construct is a possibility. And so those kinds of things so that we can actually evaluate, and especially the size of the cargo is important too. I won't get into all of the details, but some of these are important. Next part is the polymers themselves. I'm not going to go through the chemistry today because I'm sure no one wants to be bored with that, but there's lots of parts and pieces to polymers. Now, what's the good thing about that? Basically, you're mixing all of this up into a pot and using essentially Michael addition in order to drive the chemistry. This gives you a wide range of parts and pieces that can be added. I have to admit, I wish I could predict them all a priori, just look at the chemistry and figure out which one's the best. I'm going to be honest, we don't do that. Maybe eventually, once we've screened enough of them, machine learning will come into this. I heard a little bit about that from Prime, and I could certainly see how that would happen eventually. I would say right at the moment, we still use a brute force screening approach. But just to give you an idea, again, this polymer and the cargo form a polyplex. That polyplex then is nebulized, and we have a little setup, which I'll talk about in a second, where the animals go in. All of them, I would say for most of the species that we use, except for swine and non-human primates, the animals are not sedated. So we screen in animals that are not sedated, so they're breathing normally. We're not affecting their breathing rates through anesthesia. Swine, there's no way you can do that. They will squeal and scream and be very unhappy. Even if you're being very nice to them, they're still not that thrilled with having like a mask on their face. So, which maybe I can't blame them, but just the same, that's not going to work. And non-human primates, I think for anyone who's worked with them knows that's probably not going to work either. Not going to work very well. So they have to be sedated, but the other species we haven't had to. And again, the idea is that we nebulize basically our polymers, the polyplex with our reporter mRNAs. The animals breathe them, and then we honestly remove their lungs and image them in an IVUS. And so this is the first setup, and I have to admit, part of the reason this was also needed to be small, compact, and cheap is that they had to go into the BSL-3 for our COVID work, and they weren't going to come out, and so we didn't want that to be, it needed to be easy and small and not take up too much space. But basically, the animals go into these little restraints. These are a tail vein restraint. Nothing fancy about those. They go in. There's a little 3D printed nose cone, and so we've made nose cones, if folks are interested, for most of the animals that we work with. So that they stick their little heads in there, and amazingly they do that. And then here is a vibrating mesh nebulizer and a very small dead volume. And so we can go even with, we get 100-fold, 100-to-1 signal to background with even 3 micrograms. You can go lower than that. So you can use small amounts of RNA. You can, as I said, if you're only interested in maybe 2 or 3-fold. But the point is that we made about 200 initially. I'm going to talk later about the next 40 45 or so that we've made. And this is what you get out of it. And I know this seems kind of probably a little gross. These are animal lungs, but the reality is folks often want to know, like, what does this really look like? And so this is what it looks like. So here are all your pairs. And when you plot this, we can use 2 animals per group, which I know sounds crazy, but the variability is low enough that you can get statistically significant data. And if you're interested, you can go see our Nature Materials paper. But the point is that you can find losers very easily, and you can find winners. And this one right up here is polymer 76, which is one of our winners. And so I won't get into the chemistry of 76, but there was one particular element to dithiol that's actually important in its degradation, but also improved its expression. And again, that's in the paper, so I won't go into that in too much detail. But again, when we started with mice, we then went to hamsters. How do you go to a hamster? You increase the size of the tube, not too hard. New nose cones, hamsters go in here. Usually we have, again, 2 or 3 set up at a time, but this we saw expression in the hamster that looked very good. Here's our ferrets. So bigger tube. And so these we do 1 at a time. But the ferrets will, if you play with them enough, they will run in and out of these tubes and play with the tubes like it's a game. And once they get inside, you can seal them up, so to speak, in there. There's a bigger nose cone because they're kind of bigger. And I do have a picture that didn't include this, so you can see like just the end of their nose. It's kind of cute. But you can then nebulize to them, and that worked actually really well. Matter of fact, ferret uptake is great. There is expression in the upper airways and in their trachea too, but you can see it. Again, we had very nice expression throughout their lungs. NHPs, we used a pediatric mask for, and for swine, we actually use a larger mask. They are asleep, same thing with the NHPs. But the point of this was that when we looked at tissue sections and we looked at expression, what were we seeing? The mouse is actually one of our worst, and the reason is they just don't take up as much. The efficiency of delivery with a nebulizer that we've seen is between 5 and 10% of our dose in the nebulizer to the mouse. It gets better as you go to larger species. And humans should be around 70 to 80%. But as we went to hamsters and ferrets and non-human primates, but the point is we didn't see a drop off. And to us, this was really the, I guess, the aha moment that we actually might have something in our hands that would be really useful. And so I'll continue. We did some talks. So we looked again, histology. We had these scored in mice to see if there were any issues there. There were no issues in the histology. We looked at nanostring gene expression profiles. There were a few genes, but I mean literally 6 out of the 1,000 or so that we looked at that had some upregulation, and it was very low. It's also transient. So that was good. Nothing that was a problem. We looked at BioD, at least bioluminescence, though that's now changing. We'll be doing more extensive BioD studies. But so far we only see it in the lung. I ask that too for anyone who's delivering into the lung that, do you see, especially if you're coming in from if you're nebulizing, do you see any in the bloodstream? Do you see any outside of the blood? Because I'd be asking the question, are you driving inflammation, right? These particles probably really shouldn't make it there on their own. So I want folks to at least think about that. We did some histology in the ferret and in the macaque, also using a bunch of different reporters. So we used nanoluke in the macaque, but in the ferrets, both an antibody. So we do some work with antibody expression in the lung, and also with Cas13, another CRISPR protein, and they all look great. So that was I guess, I would say from a de-risking point of view, we were happy about that. We had some other folks, reviewers, who wanted to know after a dose, was there changes in animal weight? We looked at blood chemistry. Were there any changes over about a month? And the answer is no. We've done some work to look for antibodies against the polyplex itself. We haven't seen any yet, but again, this was a single delivery, multi-dosing. We do, but we're going to be looking carefully to see if there's any immunogenicity from it. But again, if you're not seeing changes in gene expression, you're not driving an adjuvant effect. If you don't drive an adjuvant effect, you're unlikely to be driving strong immunogenic responses. In a monkey, and I don't know how well anyone can see this or even read it, but the point of the issue is that we looked at a number of, we looked at NHPs at 4 hours and at 24 hours, and the point was that most of these are pro-inflammatory cytokines. There were no changes in them. There's zeros there. So we didn't see any in the blood, both at 4 hours and 24 hours. Cell populations looked good. They were all within the normal range, and blood chemistry was all within the normal range. So at least, again, I'm not saying we're done, but at first glance, it was like, okay, we did this in an NHP, we delivered, expression was comparable to the other species, and we're not seeing any major red flags in terms of problems with the animals. Not saying we're done, but at the same time helpful. This is expression in a hamster, and so you can see the control is, now I know what control's over here. And this is expression. So you can see in the larger airways, we definitely are expressing, this is beta-gal, and we're just staining, it's just IFA for beta-gal, but you can also see it throughout the alveolar space. I'll have one more picture just to kind of zoom in, and so you can see these epithelial cells are transfected, and obviously large numbers of other cells transfected. We have started looking, so the next question usually everyone asks is, what cells, have you looked at every cell type on Earth? And the answer is no, we have not. So I'm just making that clear for the moment. We actually started staining more recently with markers that we actually got from the CF Foundation, and I have to admit, your markers actually work well. I will be presenting that hopefully in the future to you all. And so we did look for keratin 5 in particular as a marker for basal stem cells. And so what you're seeing here is that's in the green, and you're seeing in the red is the beta-gal. And you can see it does appear that we are making protein in those cells. So that's a good thing. It didn't surprise me terribly because we transfect a lot of cells, but just the same, it does look like we are transfecting them. We are moving, we are going to continue to do the cell staining, but we're also moving to a 10x Xenium platform so we can start doing true spatial transcriptomics. And I think that's really, honestly, the best way. A few markers here or there does not tell you enough about phenotype, though it's better than the crappy antibodies we were using. So I would say that the new ones are certainly a lot better, but we need to look at more markers. So that's certainly for the future. So how do we improve on this polymer 76? Really mostly more work. So what did we change? We changed some of the branching molecules and our end capping molecules. And in the future, hopefully I'll be able to tell you exactly which ones, because there's actually a fun story there. And what did we do? A lot of screening. So this was our first round, and 76 is here. And you can see there are a couple that were kind of 2x-ish, and we're like, okay, that's good, but not really, no excitement. Round 2, we found one, 24H, that was about 3-fold better. And we're like, okay, that's starting to become, that's nice. And then 33C is almost 6-fold better with this reporter. And so this was really exciting. And again, what do you start to, this is what we do a lot of, analyzing lungs. A lot of our work involves, we do some work in infectious disease. And so, and I know this isn't directly applicable, but I think it helps. When we looked at 76 and even 33C, one of our new polymers, again, there's this improvement about 6x in terms of our ability to express an antibody, which from my point of view, 6-fold expression increase is good. We looked at its ability to thwart RSV. And so with 100 micrograms, this is in the nebulizer, we do get fairly good reduction of RSV. No question about that. We're looking at copy number of the RSV F protein, but we could drop it by about 8-fold and get the same, about the same data with 33C. So this was definitely exciting because it's functional, right? We're making a molecule that has to do something in the lung. Guess what we did some more of? Screening. And so out of this screen, we looked at some different versions of 33. There's some post-processing when we make some details in the production that we were looking at, and 24H, and we found 24D. And 24D is essentially the child of 76 and a number of these other molecules. And what we found is that, at least with an antibody, with the reporter, it was about 12-fold better than 76. And again, antibody expression was at least 8-fold higher. So that was, again, pretty exciting. We did some more purification of the polymer. There's some steps that we have to, and this made things crazy. So we were able to decrease the amount of polymer to RNA now, which is a good thing. So typically we're at about 25-to-1, which is not that far off if you think about like total lipid content to RNA content in an LNP. So we're in the same ballpark, but we were able to reduce it and actually improve things. So 20-to-1 actually seems to be better for 24D. We're getting great expression. We looked at Cas13, which is about 4,500 nucleotides long, fused to a nanoluke a little bit longer, so about 5,000 nucleotides, and we had about 8-fold increase in expression. And so this really, I guess my point was that it's really moving in the right direction. Particle sizes were typically in that 140 or so range. Nanometers a little big, to be honest, with 76. If we go to 24D, it's now closer to 100, which I think some people in this room will find interesting and make them happier. And then the last thing was Do we lose our species agnostic capability? And the answer is no. So this is with a different reporter, but the point was that one thing about 24D is that essentially the same dose we had the nebulizer with 76 at 200 micrograms, we get the same amount of expression with another reporter at 50 micrograms. So it was a fourfold decrease. So again, it does depend a lot on there is some variation in terms of which constructs we're using and how we're using them and the ratios, but this new 24D does look like it's an improvement. And so we're excited by that. For me, if it's more than like 4 or 5-fold with a given molecule, we're kind of excited, and I need to shut up because I've been talking too long. So take-home messages, 76 was a great starting point. We're still very excited about 76. It's a very good polymer. Combinatorial chemistry allowed us to look for new ones, new screening platforms. We found 3 new ones, 33C, 24H, and 24D that look good. We'll be doing expression now studies with the new ones in swine, NHPs, tox in rats, and I would say a whole lot more cell typing using the 10X platform. Thank you. And I thought my slides were, I had too few. Anyway, clearly not.
Yeah, that's really impressive. I was wondering if you've nebulized human ALI cultures or human airway explants.
Yes. So ALIs work very well. Matter of fact, in our flu work, we've been doing delivery to ALIs with our Cas13 construct to kill flu. But yes, we have. So we've done both from small airways, large airways too. So and with different donors. There is some donor variability, which I think would not surprise anyone who's worked with them. But just the same, we've been able to see pretty similar results across them.
Fantastic talk. I was wondering, with nebulizers, the shear or the temperature stresses.
Absolutely.
Does it change the physical properties of the polymer, or do you lose encapsulation efficiency?
So far with our polymers, we haven't seen significant, you're absolutely right. Temperature in a vibrating mesh, it absolutely heats up and with a jet, you've got tremendous mechanical stresses in comparison. So you nailed the 2 that are incredibly important. Those 2 are the reasons why we have gone towards polymers, because we don't see big changes. If you look at the Nature Materials paper, we looked at size, both pre and post nebulization, to see if there were changes. There weren't any. And we haven't seen any changes in RNA integrity either post nebulization. But I'm not going to say it can't happen, but I would say so far we haven't seen those changes, and that's why we've gone the direction we've got. That was probably the biggest reason. Because when I tried a jet with an LNP, oh boy, it was not very pleasant. I'm not saying every LNP, LNPs, everyone knows, are not monolithic single things, so I'm not judging. I'm just saying in our hands, they were more robust.
So Phil, a great talk. Just have you started to dig into mechanisms of uptake?
Yes,
do you
slowly.
feel like it is generally? Can you name specific characteristics or attributes?
I would say, what we've seen in vitro at least is they use a combination of pathways, so both clathrin and caveolin-based endocytosis, and so which probably doesn't tell you much, but at least we know it does appear to be endocytic. Beyond that, I know that some of the different parts and pieces that we've been adding now are changing endosomal release, and so it looks like we can control it, which is to me kind of holy grail. Like, can you actually control how much RNA gets released into the cytosol? But I would say that there's still a lot more on that. And that's part of a lot of what we've done is in vitro. We're doing some of those, so mechanistic studies in the ALIs, I think, will be a little more approachable. Yeah. And then, we do a lot of imaging in the lung tissue sections, and we're trying to go to super res to try to actually, destorm, to really see, because you need the high resolution to see the differences between endosomal compartments, non-endosomal. Anyone's imaged, do a CD63 stain in these cells, and there's like endosomes everywhere. And so you need high resolution to really see, are you inside, outside, and where they exactly are. But it's all doable. It's work, but it's doable.
And then just to follow on from that, so are you able to track some attributes and some of their functions, like maybe diffusion in mucus or something like that, to kind of give it some sort of a guiding principle in design?
So there's more work on mucus that's being done. I mean, we've looked through a fair, and we've even done, so just as a note in terms of mucus, we have not delivered into CF models with significant amount of mucus, so I'm not going to say we have. We've done infection models, though, that do drive mucus. Flu in particular, certain RSV strains, we know we use the line 19F mutation drives more mucus in animals. And so I would say overall, we've looked, there was a Pseudomonas model that we've used, which I didn't show data from, that we were able to deliver into animals that were very highly infected with Pseudomonas that does drive some mucus too. So I would say we've had success, but no, we need to do more work on understanding the interaction of the particles with mucus. And so hopefully we'll have design principles eventually. I can't say we are there yet at all.
Phil, really exciting. You actually answered my first question, but the second question was just about the molecule itself, or the polymer, and its how is it degraded? Do you have any understanding about its natural degradation path?
We could take that offline, but yes, I mean there's a number of different parts and pieces of it that we know accentuate its degradation. We could walk through the chemistry, but also, I mean, we've been using a part of the backbone that's been used in dental implants for a long time, and so we know that they haven't caused any tox, which is good. But there is some data from that literature on how at least that piece comes apart, and so I mean that's like the worst case scenario. If you have something in your mouth all the time that some of it's going to end up systemically, it's going to end up systemic. So we're not planning on ever doing that, but just the same, we do have some data on that, if that helps.
Hi, Alex Piotrowski-Daspit, former postdoc in Mark Saltzman's lab, actually. I noticed that you do see some elevation of liver enzymes like AST, ALT. I wonder if you can kind of comment.
They were normal.
It looked like in the non-human primate data, they're a little outside of the normal range.
They were completely in the normal range. I can show you later, but most definitely.
And then can you also comment on, it looked like a lot of your formulations were very cationic. Do you have any concerns about that?
We haven't seen any tox, and we haven't seen any changes. So I would say no question that it's charge itself is not the only reason why you have tox. And so we haven't seen changes in gene expression. So from my perspective, I mean, if we do this with PEI, I can tell you what you're going to see: a lot of inflammation and a lot of driving of type 1 interferon in particular. And so IL-6, IL-1 beta, we don't see those increases. So I would say I think charge is one aspect, but it's not the only one.
Thank you.
Sure.
Hi, I'm Sada from the CFFT lab. I have a question because all your screening cascade, the initial screening cascade is based on the mice.
It is true.
And mice are obligate nose breathers. So are these mice under anesthesia to basically circumvent the...
No, they've been naturally breathing in most cases. But when we had the swine, which are nose breathers, you could see there's actually a fair amount of throughout their upper respiratory system. You'll see plenty of transfection also occurring and in their trachea. And when they are sedated, so you do alter that some. So you do get, and same thing with the non-human primates, to be honest, also.
Okay, that's good. Thank you.
Sure.
Great. I wish to just thank the speakers for some wonderful advances. And I think there's been a lot of exciting progress just reported between last year and this year. I think we have a lot to look forward to next year as well. So